So this is Jacob Kiner for the Unit 4 presentation on gene therapies. So gene therapy was initially presented to be an alternative to standard treatments, a more sustained, long-lasting treatment that allows for continued production of proteins or whatever is needed, necessarily needed, that would replace the need for multiple treatments, infusions, what have you, during a patient's lifetime. However, initial trials in the 90s were met with some pretty disappointing and sometimes disastrous results, failing to account for various toxic genetics or reactions to this that harmed many patients and actually required a review from the a review board as well. And what the review board decided was that a refocus on some of the basic sciences of in this field, like cell biology, immunology, virology, to develop a better understanding, so that when these trials were approached again, these results like these would not be so commonplace. This allowed researchers to develop new vectors and more effective ways to introduce treatments. Some even now have FDA approval for clinical trials and have been met with some success. Initial attempts were with viral vectors. These include retroviral vectors or AAV or adeno-associated viral vectors. However, while these were effective, they were very limited to only gene addition, which treats only a certain amount of genetic issues. However, now we have genome editing, which can perform addition, like the viral vectors, as well as ablation, correction, and other tasks. And these should be performed both ex vivo and in vivo. This really took off in 2012 with the introduction of the CRISPR-Cas9 process, which has now been used in a myriad of biological research because it provides a more precise, almost scalpel-like way to edit genes. And the advancements in that have only continued since then. And these advancements have made their way actually into the medical field, and like I said before, the FDA has approved some trials, clinical trials. However, most of this is still in its infancy, and much more data and research is required before this can really be introduced to the wider, broader scale in the medical field. Current gene editing technologies include, in the in vivo section, can actually be targeted to select organs like the liver, the eye, and even neuromuscular targets, all which have seen success, especially with the eye helping to treat various genetic traits that predisposition to blindness and things like that. In ex vivo, via cell engineering, this can be various treatments include hematopoietic stem cells, which can help treat blood disorders in patients, and CAR or chimeric antigen receptors, which can actually help various cancer immunotherapies be more successful in their targets that need to be. So gene therapies have come a long way since their genesis, and still have a very long way to go, however. While the new data being presented in various studies and trials is very optimistic, and we should have good reason to be optimistic, there are still many hurdles that need to be met, and many more studies that need to be conducted before this can really hit that wide-scale application and really be approved for more general use. However, increasing involvement from pharmaceutical and big biotech companies provides funding and a wider ways to research and conduct these studies. And this should really greatly assist the research and application of gene therapies so they can be more applicable to people. My thoughts on this article: I thought this was a great perspective. It balanced both the good and the bad. It didn't just present one side. I think it was while still kind of leaning optimistic, I think it was a pretty fair and open view for both sides, and presented a very realistic look at the possible futures of gene therapies.